What a privilege to be here today with David Porter and Carl June, both professors at the University of Pennsylvania Perelman School of Medicine. Both have more elaborate titles than that and wear many hats in their careers. But both are together today because they've played a critical role in bringing a modality of what's called immunotherapy to treat cancer patients. Specifically, they've worked on a modality called CAR T therapy, which is an acronym for something long, but it's really the use of the patient's own white blood cells that are engineered and reinfused into them so that they can recognize the cancer more effectively and kill it using the powers of the human immune system. And I've brought them together today on this panel. It's a true privilege to have them in the same place and thinking about these issues and these stories together. But I think they represent two poles of an interesting sphere, which is one, David focuses more on the clinical experience, the treating of the patient, caring for the sick in day out behaviors in the hospital. And Carl works more in the laboratory throughout his career, focusing on inventing new science that can be brought to the clinic for people like David to make use of. And it's really their partnership together at the University of Pennsylvania that's made possible the FDA approval of the therapy I mentioned. And so this therapy, CAR T therapy, is a proven therapy that the FDA has reviewed and deemed safe and effective and fit for sale in the United States. And this therapy is used in treating certain kinds of aggressive lymphomas and pediatric and young adult leukemias. And we're hopeful collectively as a field that this therapy can go beyond those indications and help more patients. My own background, I met both gentlemen in my training as an oncologist at the University of Pennsylvania. After that, I worked at the FDA as a reviewer. I worked in venture capital, helping finance technologies like the one we'll talk about today. I started a biotech company, and today I work for a large pharmaceutical company called Eli Lilly. But really, I'm here as a fanboy, as a spirited enthusiast of the work of these two gentlemen, and I wanted to reveal again their life stories and their professional paths to a larger public. And that is my motivation for being here today with them. So with that, I'd love to ask you both the same question and have you answer it. So why did both of you enter medicine in the first place, and what else was in contention, and what were some of the early choices that led to this career for each of you? David, perhaps you first.
Sure. Thanks, Josh, and thanks for having us. I think we're both disappointed we're not in Provincetown and we're doing this virtually, but it's really a nice program, and I appreciate the invitation, and it's nice to see you again after all these years and knowing all your tremendous successes. My career path was somewhat interesting, and I look back at where I am now, and I fascinate even myself that some of it was predictable. I was in school, and I was in college, and I was a biochemistry major. I was always interested in science, and I became fascinated for various reasons about regulation of gene expression, how cells control genes that make them what they are. And in fact, cancer cells become cancer cells because their genes don't work normally. And I was starting to think about, even in college, if we could understand how genes are controlled and regulated, perhaps we could understand what makes a cancer cell, and then we could manipulate it and maybe treat cancer. So I got involved in college with some research being done in oncology, and I was very interested in the basic biology of cancer. Didn't really know what I wanted to do after college. I went and I worked in a laboratory at Harvard Medical School right in the dawn of the era of molecular biology, where people were just understanding how to isolate and manipulate genes, how to understand what they did. And I was fascinated by the whole idea and thinking I was going to go be a PhD scientist. But I was unsettled. I didn't really know what I was going to do with it. And I don't remember all that many details, but I have a crystal clear memory of going to a lecture. I had access to these lectures of these Nobel Prize laureates and world leaders. And somewhere around 1982, I went to a lecture by a man named Y.W. Kahn, who was in San Francisco, who was an expert in sickle cell disease, and gave a lecture about how someday we're going to be able to cure sickle cell disease by putting new genes into bone marrow cells and fixing it and curing it. And it was like a light went off that this is what I wanted to do. I wanted to be able to do gene therapy, maybe for sickle cell disease, but maybe for cancer. And I became interested in hematology and oncology. In fact, I still have a copy of my medical school application. At that point, I decided I would go to medical school and learn the medicine, but maybe be a scientist. And my essay for medical school was about doing gene therapy to cure cancer. I went to medical school and I got interested in doing bone marrow transplant and hematologic malignancies, blood cancers, because bone marrow cells, you could easily, and blood cells, you could easily take out of the body and you could manipulate them. And it was something I was always thinking about. What we're doing now with the CAR T cells, the chimeric antigen receptors, are in fact taking out human immune cells, genetically changing them so they can now recognize and attack and kill cancer cells, is very similar to what I was kind of dreaming about doing in the early 1980s. It sounds a little bit like, gee, my career was laser focused to get to this point. That's not exactly how it all panned out in the interim. Bone marrow transplant therapies, and then I think the explosion of my career came when Carl came to you. I think the most brilliant person I've ever met, and the rest is history, as they say.
Carl, did your medical school application predict your career also?
No, it didn't, Josh. And first of all, just congratulate what you did at Lovo. It's an amazing story that we need to talk about at another meeting. But there's a lot of parallels with my background and David's, and some interesting distinctions. And I thought, I graduated from high school in the height of the Vietnam War in 1971, and I was admitted to Stanford. My dad had been a chemist, and I thought I was going to do the same thing. There'd never been a physician in my family. And then I got a very low draft number, which meant I was going to go to Vietnam in the rice paddies. And instead then what I did was never went to Stanford, but went to the Naval Academy, where at least I thought when I graduated, I would be as an officer, not in the rice paddies. And then the Vietnam War got over, and the Navy then, and they had a pre-medical program. I got interested in biology and went to medical school. And I really loved clinical medicine. I thought that's what I would do the rest of my life. But I graduated from medical school a year early and was allowed to do a fellowship at the World Health Organization. And a big part of my career then has been infectious disease, even though I'm a board certified medical oncologist like you all. So I spent a year working on malaria and then got interested in research, and gradually went from reading the New England Journal of Medicine to reading science. I got interested in the lab work, but because I was in the Navy, I had to spend 10 years working on infectious disease, because little known to most people is that all the congressionally directed cancer research has to go through NCI. And I was in, pardon?
The National
National Cancer Institute. So the National Cancer Institute then funds cancer research. I was a board certified medical oncologist. I had really been fascinated in transplantation, BMTs, just because that had just come out, and David and I were in parallel in that part. And then But when I went in and started my first lab, I had 10 years of obligation to the Navy at the Office of Naval Research, and I was only allowed to do research through the DoD in infectious diseases. So for 10 years, I worked on HIV, and I backed into gene therapy that way. Our first chimeric antigen receptors, which is the abbreviation for CAR T cells, our first CAR T cells were given to HIV AIDS patients because there were no good drugs back then in the 90s. And then, so that gave us a lot of experience, sort of an out of the box thing that the other oncologists didn't have. The fact that I had to spend 10 years doing HIV research. Then, as David mentioned, I came to the University of Pennsylvania in 1999, and that was like a marriage in heaven. The two of us, he was the world's expert in getting T cells in the setting of bone marrow transplants, and I had been working on genetically engineering T cells in the lab, and then we got, I think, the first ever grant to do a CAR T cell trial in HIV, in cancer patients, leukemia. That happened in 2004, and then that led to the rest of the story.
Did you guys meet right away in 1999?
Yes. I mean, we'll see what David's recollection are, but I mean, we, in the recruiting process, so I moved in 99, 3 senior people moved with me so we could establish a team for gene therapy and cancer. And David was the go-to person to do this. And it was just perfect. He could concentrate on the clinical aspects and I could work on the lab aspects.
David, do you remember the first meeting?
Oh yeah, I came in 1996. I predated Carl at Penn. I had been in Boston, and I came really to develop a bone marrow transplant program. And I think in those 3 years, we had developed a really good program. Bone marrow transplant is something that we use to treat patients with leukemias and lymphomas. We give them high doses of chemotherapy, we kill off their bone marrow, and then we give them bone marrow from somewhere else. But it's also immune therapy because the way that it works is we also, with the transplant, give them a whole new immune system. Those new immune cells, those T cells, can help fight that patient's cancer. And we did a good job in those 3 years of developing procedures to make it better, to make it safer. But you can only go so far. As a clinician, you can take really good care of patients, you can do some innovative things, but you can only take it so far. When our cancer center director called me and said, boy, I just heard Carl June has agreed to come and his lab is coming with him, it opened up a whole new world of immunology and biology and things that we could then offer patients by collaborating with Carl and working together. So for me, it was a giant leap in the abilities that we had here at Penn to really take care of patients and offer them some really innovative, novel approaches that we never thought of before. So I remember it really well.
When people hear the term transplant, it's a little hard to visualize. When you hear lung transplant, you can imagine what the moving of lungs from a donor to a recipient looks like. But what does it look like for bone marrow transplant?
So I will take that, but a bone marrow transplant is something we use right now primarily for people with blood cancers, leukemias, lymphomas, multiple myeloma. It generally involves treating a patient with very high doses of chemotherapy, sometimes radiation, that is designed to get rid of all of their bad, sick blood cells. Blood cells come from the bone marrow, and so it's made to get rid of their bone marrow cells, which then make their blood cells, which is where their leukemia is. You use the chemotherapy and radiation to wipe out all their leukemia. It wipes out all their bone marrow, good and bad, and it leaves a patient without bone marrow. Bone marrow is needed to make blood cells. If you don't have bone marrow, you can't make blood. If you can't make blood, you can't survive. So what we do is we treat the patient with these high doses of chemotherapy, radiation sometimes, and then we collect bone marrow cells from a donor whose immune system is closely matched. We collect the bone marrow either by taking it directly out of the bone marrow. There are now ways you can collect bone marrow cells right out of a person's bloodstream. It comes in a bag. It looks just like a bag of blood, and we infuse it into a patient's bloodstream right through an IV. It's very smart. The bone marrow cells find their own way from the blood into the patient's bone marrow. They sit down, they start growing, and in about 2 weeks, they start making normal blood cells again. So for the patient, it's not surgery like you think about with a lung transplant or a heart transplant. It really is an infusion. That said, it's every bit of an organ transplant like any of the other organ transplants we think about.
So Carl, why isn't that good enough? If you're replacing a normal immune system into a cancer patient who presumably all the cancer is gone because of the chemotherapy, why isn't that good enough? And why do we need research like yours to take it to the next level sometimes?
So that's a great question, Josh. I was fortunate to train at the same time David did. Bone marrow transplantation really happened in the early 1980s. He was at the Dana-Farber Harvard system, and I was at the Fred Hutchinson Cancer Center. And the first transplants that were successful were done then, and they required really very high doses of chemotherapy and irradiation to leukemia patients in order for the bone marrow from the donor to take in the patient with leukemia. And so there was lots of toxicity from that. It was so-called super lethal chemotherapy. And then the bone marrow that came in from the donor would so-called rescue the patient so that they wouldn't die from the radiation. And then they would get the benefit that could sometimes cure their leukemia because of that very high dose of chemotherapy and radiation. But there was a huge side effect, and this is actually one of the major contributions David did at Penn and well at Harvard, which was the patients could get graft versus host disease. And that actually was where the T cells in the donor would kill cells in the patient, but they also killed leukemia cells. And that was a huge understanding in how it worked. But that side effect was a major toxicity. And so what we worked on, and what a CAR T cell then is, one way not to have to do a bone marrow transplant, because you can use the patient's own T cells, not give them the donor cells, and then you can't have graft versus host disease. And then the CAR T cells from the patient then are the ones that do the heavy lifting and kill the leukemia cells. So that was the major difference, not having to do that. And so we actually give CAR T cells now to people who've already had a transplant, and as hopefully they won't ever have to have one. So right now we're in this transition period that way, but that's the goal.
I think the last few minutes have laid a really solid foundation of the science of what you've done together. We're going to go back to it, but maybe we just take a quick tangent to the emotional and human side of taking care of patients with these diseases. And David, I'll start with you. And I know every day you're in the patient rooms on rounds and during the day at times. And what's it like treating patients really who are threatened, their lives are threatened by this disease? And how do you hold up emotionally after so many years of doing it? And comment, please.
Yeah, Josh, that's a good question. It's a hard one. A lot of people with leukemia and lymphoma really are facing life and death. And we do deal with that every day. I think from my standpoint, there are good days and there are bad days. There are times when no matter what we do, it's not good enough. But there are people who have incurable diseases or who otherwise had incurable diseases that we can now help and we can fix and we can cure. People say to me all the time, gee, when are we going to cure cancer? And my answer is, we do cure cancer. We cure cancer every day. We just don't cure it often enough. But the ability to take care of a patient and to go into the room and say, you know what, you are in remission, your cancer is gone. It is one of the most rewarding experiences I think, in life, that it gets you through those times when you um, uh, don't have enough to do for a patient. I saw a patient this week who was on their 24th anniversary of their bone marrow transplant. They had lymphoma. They were dying from lymphoma. They had a bone marrow transplant 24 years ago. She comes back to see me every year. We rarely talk about medicine. I see pictures of her kids and her grandchildren, just had her first great-grandchild. You know, the rewards from that are unimaginable and difficult to explain. You know, a bad day is certainly when things don't go well, that you can do all the right things, and the cancer is very smart. Cancer is often smarter than we are. No matter what we have, what we apply, it doesn't work. I'll tell you, the worst days are when we don't have anything to do. We don't have something new to try. We don't have something novel or experimental. When you have to say to a patient, there's nothing left to even try. And those are the hardest days. You go on because there are other people who need help, and there are other patients who still you have to take care of, and you have the potential to fix and to cure and to make them better.
On the hard days, does the hardness follow you home?
Sometimes. It's, you know, impossible that it doesn't affect your life. This is as you can imagine, and as you know, it's not a 9-to-5 job. It is what you do all the time, and it follows you home. Yeah. There's sadness on some days. There's frustration on some days. I have a wonderful family. I have a 14-year-old son, Noah, who just started high school today, and my 9-year-old daughter, Sophie, my wife, Kathy. I go home, and they are tremendous support. My colleagues here at work all go through the same thing. We all commiserate. We all talk. I think that's sort of a natural friend and support group. We all go through the same things, and we understand it. And then you have other friends outside of work and other family that help get you through some of those frustrating down days. I think in the end, you know, you do absolutely everything you can. You try and do better. You have something to offer patients. And sometimes the job is helping them through really difficult, frustrating times, and even helping them and their families when they are dying and medicine isn't good enough. And it does follow you home, and sometimes it's hard to shake until the next day when you have more patients that come in and are looking to you for help, and you have to be able to help them.
Carl, in your career, you've been in the clinic as well, and you've later spent more time in the laboratory. How does your relationship to patient care change over time, and how does the personal patient experience affect your work today?
Yeah, well, it has changed. Until I came to Penn, I saw patients all the time. I'm still credentialed, but at this point, I jokingly say to my family that my medical degree, the MD, stands for mouse doctor. So I don't see patients other than refer them to people like David who see them every day. And I have, there's a huge advantage to seeing patients and knowing what the problems are in a clinic. I can remember, it's seared into my memory from the early 1980s, those first bone marrow transplant patients, and some of them who made it and some didn't. And I went through this time, where I really enjoyed pediatrics and internal medicine. And I decided I could never be a pediatric oncologist, but that just was, I was unable to stand that, those issues. So my hats are always off to my collaborators who are pediatric oncologists, and now, and how they do that, I'll never know. It's not something I can do. So as an adult oncologist then, I mean, there's great joy in what David said, your successes, and then other times when it's not that. And I think now the general public understands a lot more of that, this burnout issue you can get if you're not careful. And, you know, with the COVID epidemic, there are physicians now, you know, pulmonary intensivists and infectious disease specialists who are now saying what oncologists did all the time. I mean, the infectious disease people have not witnessed this since the AIDS epidemic early time. And then, you know, basically they were called in on consults, but now they're seeing patients who are healthy and dying, just as the oncologists do. And so that is a problem with the medical profession. And some people have different gifts as physicians on this issue of compartmentalization. You know, can you go home and have it not destroy you, some of those burdens you carry, or not? And some people are much better at that. So that's something you have to have. And the nice thing about oncology is that there are some spectacular successes, as you know, and other times not. But those, you know, I always talk about the sports analogies. You know, baseball players go up, and if they get a hit every 1 in 3 times, meaning a failure, they're a failure 2 out of 3 times, they're okay. In fact, they're at the top of the profession. You know, we have some, it depends what cancer you're seeing, a success rate and a failure rate, and our motivations are to make that, you know, like free throw shooting, where most of the time we make it, and there is success in a cured patient. But I have memories of the patients we first treated with CAR T cells, you know, and I share those with David, and it's, you know, they're now beyond 10 years of patients who look like they're cured. And that really gives you motivation to go forward into areas where we're not successful.
Josh, can I editorialize for a minute? Can I jump in? You know, what we do here at Penn is, in the very best sense of the word, translational science, translational medicine. I think that we translate some of the science and things that Carl does in the laboratory into clinical medicine. And if you think about translational science, it's this big spectrum that you have very basic scientists on one end and people who just see patients every day and take care of patients. And the translational work happens in the middle. Carl is on this side. He's not the basic scientist. He is the translational scientist developing new therapies and science that cross the line over into the clinic to give to patients. And with the CAR T cells, you know, we haven't gotten into some of the outcomes, but I will just say that we are curing patients with cancer who are incurable and were destined to die in a few weeks if they didn't get these. And that was developed in his lab. So he said to you that he doesn't see patients. I'm gonna correct him. He doesn't take care of patients. The highlight of some of my patients' journey through this whole thing is they got to meet Carl June. They come in and I meet Carl, and he has come over to the clinic. We have had ceremonies. I have the most fantastic pictures of these patients standing with the guy who invented this therapy that cured them when they were going to die. And I won't talk about, you know, well, I will talk about it. I don't know that I've seen Carl meet a single patient where he doesn't get choked up, where the emotion just pours out. So don't let him tell you he doesn't see patients and get the impression that he's stuck in his lab coat in an office on a computer or in a laboratory. Certainly patients see him, and boy, what an experience.
Can you guys maybe pick a specific story to tell about a patient where things look bleak, and maybe even there were barriers to even bringing this exciting possible therapy to them, and you overcame those barriers, and tell us how it affected the patient? Does one come to mind?
Carl, you want to talk about Emma?
Well, I can talk about a pediatric. I think you could talk about patients 1, 2, and 7.
Yeah.
And,
all right. Well,
and, you know, yeah. The adult ones went first, and then we had pediatrics.
Yeah. So we did. We started treating adults first, and this was in 2010. And And I remember the very first patient. He had no treatment options left. He had a disease called CLL, or chronic lymphocytic leukemia. It's a cancer of the blood cells. He had been through many different therapies. None were working. There was nothing left to treat him with. And
How old? Who was he? How old was he? And what was his background?
So I can actually tell you who he was. His name was Bill Ludwig. And everyone's gasping because you can't use patient names, and there's all sorts of privacy concerns. Bill Ludwig's been written about in the New York Times and in many other areas of press, and has been very public with his story. And Bill was running out of options. This therapy had just been approved for a clinical trial, and he was the first patient to be appropriate and to agree and volunteer to get this therapy that's never been given to a human before. We gave him the cells. We were standing over the bedside as he was getting the cells, just watching him like crazy people, and nothing happened. Until about a week and a half later, when he started to get sick. And in fact, he not only got sick, he became critically ill. One of the reactions with these CAR T cells is what's called a cytokine release syndrome. Cytokines are these chemicals in the body that cause that result from inflammation. They cause this massive outpouring of fluids, high fevers as high as 105 degrees, a fluid leaking into the lungs, making it difficult to breathe, dangerously low blood pressure. And Bill had to be cared for in our intensive care unit. And there was concern that he was going to die from a side effect of the cells that we gave him. He got better. We treated him very aggressively, but I think in the end, he just started getting better about 3 weeks, 4 weeks after getting the cells. And I have two very specific stories about this. As he was getting better, his blood counts, his blood tests were now looking normal for the first time in years. Never had normal blood counts. He had swollen lymph nodes. People with this type of leukemia have the leukemia cells growing in the lymph nodes. You can easily feel, people say swollen glands, but swollen lymph nodes, we couldn't feel them anymore. They were gone. And the real way to look for leukemia is to do a bone marrow test. It's with a needle in the back of the hip. You take out a small sample of the liquid bone marrow and a piece of the bone. And we did a bone marrow test on Bill about 4 weeks after the T cells were given, and there was no leukemia. There's no precedent for that. There was no treatment that should have eradicated his leukemia. We were so excited to call Carl. Carl, it worked. We did this. There's no leukemia. And his answer was, oh, that can't be true. There must be a mistake. Go back and do it again. And so we had to go back into his room and very sheepishly say, gee, you know, we're not sure about the results, and we have to do this test again with another needle. And we put him through another bone marrow test, and lo and behold, there was no leukemia, and he was in remission. It's a very similar story with the second.
Like, Carl, why were you skeptical on the phone that day?
You know, mostly when you have an unexpected result, it's due to a mistake in science. I mean, usually you fail. And so to have, I mean, we had been doing this for a very long time. And there was about only, and we haven't talked about this, there was no industry then. There were maybe 4 other academic centers trying to do this. And they had all tried this in various kinds of, and it always failed. So what we had done is made some design improvements in how the CAR T cells were working, but honestly, they didn't work that much better in our mice where they had experimental leukemia. They worked better, and we published that. And then David tested it in the first human, and it was like a home run compared to a blunt single. It was amazing. Yeah.
Were you guys both scared that week he was in the ICU?
Yes. I mean, we had, you know, there have been highly publicized gene therapy deaths. And because you were modifying the genes of these cells, that means the DNA, the code to put in the part that writes for and specifies that there's this chimeric receptor molecule that kills leukemia cells in there. So it's very highly regulated, and so we were worried that if someone died on that, that would be the end of the trial, because that's how it had been in the past. And you know, in all honesty, we had no idea what was happening. This cytokine release syndrome and the high fevers that David described had never been seen in humans. It didn't happen in our mice preclinically, you know, so it only was discovered when we saw a patient there at literally, you know, in an ICU who had, you know, already written his funeral instructions and so on. I mean, we thought it was time for, you know, last rites.
David?
You know, Josh, there's another phenomenon too. Yes, we were anxious and scared, and you know, there's also a difference between a patient getting critically ill and you're treating them and the treatments aren't good enough and they just don't work, and that's just awful and horrible. There's a difference though when this is happening because something you did. These were cells we made and we infused into this person, and now he's getting so sick. Like, what did we unleash? What did we do? We kept asking. And you know, thankfully the story has a good ending.
So when things started to get better and exciting, and you called Carl expecting to high five over the phone, were you surprised at Carl's reaction?
Boy, yeah. What are you talking about? Like, we're so excited, like you're dampening the party. But no, you know, surprised in the end. It was, oh yeah, that's right. That's the right thing to do. And that's, you know, that's the other reason you have a team. Yeah, I think, right, you're supposed to be excited and you just want to hear, well, I don't believe it.
But we were talking, so how did the patient react to the news?
Well, yeah, I think for that news that we were going to do it again, he didn't care. He just wanted the answer. But when we got to tell him that there was no signs of leukemia, it's you know, just the tension disappears. There were tears. His wife was with him. It was, you know, as I say, unprecedented, and it's not something we have an opportunity to do often. That said, I remember the first few patients. You know, what we knew is that we killed a lot of cancer cells, and we were learning that in real time. I mean, we were measuring it, and we learned that we could kill with one infusion of these immune cells, we were killing somewhere up to about 7.5 pounds of tumor. But this was 1 month later, and we had no idea it was going to be sustained. So I will be honest, and we tempered it. And I often get accused of being very conservative as a physician, but the discussion was, you're in remission. And you know, remission is kind of a wishy-washy medical word that we use to say we don't see cancer, but we don't know if it's all gone. And we had no idea if this was going to be sustained. So this wasn't a, you know, you're cured, fine, you know, it's all done. This was, things look great, they're going really well. I'll see you next week, and we will keep an eye on it. But as Carl said, that see you next week is now 10 years ago, as of the last week in July. He is 10 years in remission. And the vast majority of people destined to relapse have their leukemia come back very quickly. You know, we believe that these patients now 10 years with cancer-free are almost certainly cured. And so the excitement builds over time on some level.
So Carl, we're already talking about the success, but I know you went many years in your career where success wasn't at hand. And help us understand how you persevere in the face of that kind of failure. I get, failure may sound strong and harsh, but what's that like? And were there dark moments where it could have gone another way, or maybe you moved on?
Yeah, for sure, Josh. So I mean, I mentioned that we got the first ever grant to try to treat patients with leukemia with these kinds of cells. And it's really expensive to do this because there was no industry at this time. And you have to imagine back in 1999, when we started this, in 2001, we first started thinking about how to do it and then treating mice. There was no biotech. Usually when you have something you've done in the laboratory, you can then work with a biotech or pharmaceutical company, and then they can do the tests that the FDA requires so that you can make the new drug, the so-called investigational new drug, or IND. And in this case, we had to make it ourselves. And it was the first time, and that meant we had to make a virus that would change the cells. That had very high standards, and we had to, the whole application was more than 1,000 pages long to the FDA, Food and Drug Administration. And that was done between 2008 and 2009. And then you may recall what happened in 2008 to 2009. There was a collapse of the whole industry, due to Wall Street issues. And, we had a recession, and we had major donors that were supporting our trials, and we had the Leukemia and Lymphoma Society supporting our trials, and their donors dried up during that recession. And I had to fire many people, unfortunately, and almost close up shop because we couldn't support the team that was required, to make and conduct a trial that led to Bill Ludwig. And so when we actually got those results with Bill Ludwig, we had enough of that virus to treat 3 patients. And then we ran out. And that virus back then cost $500,000 to make. And it was like time to fold up. And for me, it was a decision of just going back to basic science research and forget about trying to make, this translational space and treat patients, as David mentioned. And so David and I met in a coffee shop back in February of 2000. Let's see, we treated Bill in 2010. It was 2011 in February. It was a deep, dark winter of 2011. And because we'd run out of money, and we decided, what would we do? We treated 3 patients, and all 3 had responded, which was really unheard of. And we didn't have the money to go. So we decided, because our trial said to treat 14 patients. That was the phase 1 trial we'd done, but we couldn't complete it. We decided to do something very unusual, which was just to summarize the results of the first 3 patients where they'd all 3 responded, and at least get that out in the literature. So we submitted the paper, and then it was published in the New England Journal of Medicine as a case report in August 11th of 2011. And our lives, for me, I can say for sure, completely changed that day. And I can say that I'm pretty sure confidently the same thing for David, because our funding worries were over then, because it got a storm of public attention. Pharmaceutical industry woke up and said, well, maybe this could be something other than just an academic, curiosity. And so we went from closing up the shop to this overnight thing of success, and how can we treat more patients? How can we extend it to pediatric patients, and so on, based on the success of 3 patients. That was it.
That morning in the coffee shop, I mean, you guys had spent your careers trying to get to this moment scientifically, and now there's this mundane thing of money that's holding you back. I mean, what were the emotions of that morning?
Dave, do you want to mention?
Yeah, it's super frustrating. Helpless, I guess, is probably the best word. Frustrating, helpless, that you have this thing. The science is the most exciting thing that we have seen in ages. It was a home run. It wasn't a single. And people's lives were potentially in the balance, and there was no money to do it. And you feel helpless. You try and you write grants, and Carl said, well, we should publish this, and I'll admit, you don't publish a single patient. Nobody would believe it. Nobody would be interested in it. But boy, it was very, very forward thinking. It turned out to be the right thing to do.
Yeah, in part, immunotherapy was not a new idea when David and I started working on this. There had been, the bone marrow transplant field had opened up the concept that T cells could kill cancer, but the pharmaceutical industry had given up on it. But we had, what was different about Bill Ludwig was, because those cells had their DNA was changed, that the code, we could follow them in the patient, by just taking a blood sample. And we could show that they in fact had divided, and that 7 pounds of tumor that David mentioned that Bill Ludwig had, that there were also at the peak of the reaction, meaning 2 to 3 weeks after we treated him, there were also 7 pounds of CAR T cells in the patient. So a small amount that we'd infused, literally just a little pellet like the size of a pencil eraser, had become pounds of cells. So the patient himself, had become a bioreactor. That was a completely novel concept, that the patient could grow their own cure. And we now know, David didn't mention, but July 30th, when 31st Bill Ludwig came back 10 years later, our lab studies show the patient, he still has CAR T cells. So the term a living drug was coined. We'd never had that before. Now we have the concept that you can give a single infusion and the cells can be parked in the patient. And now we know for a decade, and be on patrol then and still be able to kill recurrent tumor cells. So it was a paradigm shift that's rarely seen. With the concepts were there, but it just took all the right technical details. And then we had this amazing change between something that actually, for the first time in my career, it worked better in the patients than it did in the mice.
So your careers have collectively touched, have involved government, the National Cancer Institute, the Navy even, have involved the university system, have involved, private insurance, have involved the pharmaceutical industry, the biotech industry, philanthropy. Over the last 10 years, has it changed your views over, in the public sphere? Where should funding come from? Who should pay for this stuff? What's the best or right way to do it? What's fair, but what's appropriate? Do you guys have views on these very complicated topics?
I'm going to start and then let Carl go, because he can be much more articulate, I think. But you did say, Josh, in the last 10 years, and I think that's a good timeline. There was a time when academics and industry on to mix academic research with industry funding, with industry collaborations. There were all sorts of concerns about ethics, and I think there was a time when that was not considered either acceptable, or there was a certain amount of distrust that was there in our field as a medical community, maybe even as a scientific community. But I think that has evolved. You mentioned a whole lot of places that we have touched upon. Everything's different, but I think what that highlights is this is a big collaboration to do this big kind of work. To develop these incredible therapies requires collaborations and interactions from all kinds of different resources. I'm going to let Carl expound on that, but I think that has changed dramatically in the last decade.
Yeah, it certainly has. Initially, all of new basic research for cancer really was funded, and for decades, as you know, Josh, by the National Cancer Institute. And then traditionally what would happen is advances would be picked up by and licensed out from universities into pharma. And then, the FDA approval process would begin. And so that was our pipeline. But I can tell you, I have, when you submit a grant proposal to the National Cancer Institute, it gets judged and then voted on, and then it gets, right now, maybe 10% of the grants get funded. And you get back the peer review, which is other oncologists look and say the likelihood of this working, and they're called pink sheets. And I put in with David 3 different times grants to the National Cancer Institute, and I got pink sheets back, and they all said, no, this will never work. It's, you know, these CAR T cells, they've been tried before. This is, you know, a pipe dream. So we could not get funding for that trial for Bill Ludwig from the National Cancer Institute. And where it came from was philanthropy. So one lesson we've learned is that, you know, long shots, and so they're more willing to take a risk with longer view through philanthropy and there's a variety of foundations that do this, than the government funding agencies. And then, and there was no industry for cell and gene therapy at that time in early 2000, and then by the time we treated Bill in 2010. Now there's been over $10 billion invested in the US just in CAR T cells. It's amazing, between Gilead, Bristol Myers, et cetera. And so now there is a rich pipeline happening, and it's what David was, it's more like standard drug development now. Cell and gene therapy is a partnership between pharma and biotech, and it comes back into where we're working at the University of Pennsylvania and making the next generation ones. But I still worry that the long-term projects that, you know, you want to have some, you know, 3-pointers and not just always shooting layups. And right now, our government funding agencies are very conservative, and they're mostly looking at funding layups. We need to have systems that will make sure to take enough of our funding from the government and from pharma on long shots, because in general, they don't take long shots. And venture capital will do that, and that's a big, but you know, they only look at like usually 3-year time frame. So there's a lot of issues now on how you have a balanced portfolio between what the government funds, what venture capital will fund, and then what pharma and its deep pockets will fund.
If they're young trainees in medicine or PhD scientists listening to this, and they're choosing a career path now, what would each of you say to the emerging professional about emotionally how to approach a career and how to make choices that have the biggest impact?
I think it's certainly a personal decision. What excites you? What are you interested in? It has to be something that excites you every day. Certainly in medicine and science, I think you highlighted well with some of the questions that this is, this is a difficult field with a lot of emotional concerns. It has to be something that you believe in. And gets you out of bed and excites you to get you through it. And it is exploding. I think about oncology and the new cancer therapies and the changes that have been developed over 2 or 3 years, let alone over 10 years. It is absolutely phenomenal, the progress that has been made in cancer therapy and understanding cancer biology, how to target abnormalities in cancer cells, as you know as well as anybody, Josh, how to use targeted therapies to kill cancer. We are making huge strides. People are living better, they're living longer, they are being cured when they couldn't be before. And so if that's something you find exciting, there is no better place to study and work right now than in that field, as far as I'm concerned.
Yeah, I mean, it's hard to add to that from what David said. I think it used to be, the one thing I would add is it used to be that one had to make a decision, either I'm going to be in an academic medical institution or I'm going to go into pharma. And there was very little interchange between them. you kind of had made the decision, either you went, quote, to the dark side, the academics would say, if you went in the farm. And now it's very different. There's both interchange back and forth. We depend on each other, but also you can, and your own career shows this, you can go back and forth now between them. So I think the young people now have the amazing thing that there's much more flexibility. There's much more jobs in this area than there was before. And that the tools that we have in basic science pave this with a very wide road now of, we're just at the beginning of this, is what I think. So I just would be very excited. I mean, I have 5 children now, and the first one's a PhD academic person, the second one's an MD, so they made decisions to be either basic or clinical, and it's just such an exciting time for them. And so I think, you know, once we get behind this pandemic, you know, it's going to be, we're just off to the races with the real race on cancer cures.
Fantastic way to end a really special hour. I'm grateful to you both, David and Carl. Thank you for devoting some time to us today, and take care. Godspeed.